All right, y'all, this is Mr. Labonte. I'm going to start talking about acquired immunity. Two things we want to think about when we talk about acquired immunity is we want to think about the antigen. So this is the whatever is in the body that a body's reacting to. And then we want to look at the molecules that are binding to the antigen. So first of all, you have the antibodies. So these are these molecules. They're designed to bind to antigens. They can do two things. They can destroy or disable the antigen, or they can warn other cells that the antigen is present. These antibodies are created by random mixtures of DNA. So this is DNA here. So this is one chain of DNA. Here's another chain of DNA. These two different sequences of DNA combine to make an antibody. Some of these DNA sequences, like this part here, are fairly stable. They don't change at all. But then there are these regions which are highly variable regions out here on the end. And they're made by randomly selecting different, say, V, D, or J parts of the DNA and putting them together. Because you have so many different selections of DNA that can be added together in random ways, you can get 10 to the 14th different ends on these antibodies. So that's a huge amount of differences in antibodies. So pretty much any way that you can put different proteins together, we can make an antibody to fight it and to figure that out. So this is all done randomly by selecting these different sections of DNA. Antibodies, this is what's called an IgG antibody. So this is kind of the classic one. This is the most numerous antibody that we have, but there are IgA antibodies and IgM antibodies. This just discusses all the different ones. They have slightly different jobs. Okay, so we're randomly combining all of this DNA, right? And this DNA, as it's getting randomly combined, is making antibodies. But there's a problem with this, which is we could be making antibodies that will bind with our own body and can attack our own body. And so what we do is we randomly put these together and then we test them to see if the cells that have them will react to our own body. And then if they do react with our own body, then we have to kill them. So that's called self-tolerance. It's just a limit series. So here, this is the clonal selection theory. One, you have a B cell generally, but it could be so you have a B cell that is has no antibodies. It randomly generates all of these antibodies, and some of these antibodies react to the human that they're in. So if that's the case, they get destroyed, and then all the rest of these different ones go out into the bloodstream and are floating around, and they're not doing a whole lot, and there's not a lot of them. But one of them happens to be perfect for a particular antigen, and once that starts to happen and once they start to bind, then you start getting replication of that particular cell. So this is how this happens, and I'll go into a little bit more depth on that. This is an image that I like a lot because it shows the distinction between T cells. T cells develop in the thymus, and B cells, which develop in the bone. So that's why they're called B and T cells. And they have generally different jobs. B cells secrete plasma, and they secrete antibodies and that attack antigens. T cells kill different cells, so they can kill infected cells, but they can also kill pathogenic cells as well. And then T cells also make these helper T cells, and we'll talk a little bit about helper T cells. I'm going to go to a slightly more detailed drawing of this because I think it helps show these things. And this is from these presentations that I showed you in the last class. This is obviously really busy. There's a lot of things here, but I'm just gonna go through. I took some sub slides of this, and I'm gonna go through just a couple different ideas. So remember, all of these cells come from the same stem cell. This is on the lymph system, so these are the lymph cells. We've talked about this. If these cells, be they T or B cells, react to us, to a self-antigen, then they're killed. If not, then they're allowed to survive, both the B and the T cells. T cells have T cell receptors, and B cells have these antigens, antigen receptors. So we're going to start and focus on T cells first. So first with the T cells, there are two different types, and it depends on this protein that accompanies the antigen. So it could be a CD4 or a CD8. And the difference here is that CD8 T cells are going to bind with an infected cell. So this cell got infected by the mechanism that we talked about. This is the MHC1 protein. They chopped up the bacteria or whatever was in it, and they are now presenting this little chunk of the bacteria or this antigen to the T cell receptor saying, I'm in trouble. When a T cell sees that, it becomes activated, and then it becomes a cytotoxic T cell. It also will replicate, so you'll get a bunch of these cytotoxic T cells, and then their job is to go around, hunt, find these infected cells, and then destroy them. So that's the job of CD8 T cells. They find and kill infected cells. CD4 T cells have a different job, and we've talked a little bit about how this works in general, and that is to say, now we have a cell like a phagocyte. So this is going to engulf. So this is a white blood cell, the innate immune system. It engulfs a bacteria, destroys the bacteria, and presents that bacteria. But now that bacteria, or chunk of bacteria, virus, or whatever, is being presented on an MHC2 protein. That binds, and activates the CD4 receptors. So this is a different thing. These guys intentionally got the bacteria antigen in them. The ones up here in the CD8 did not intentionally get this in them. And then the CD4 T cell becomes activated. It will replicate, so you'll end up making more of these that are activated for this particular bacteria. And it's then going to alert natural killer cells and all macrophages to let them know that this particular antigen is what they should be looking for. So that's the role, one of the roles of the helper T cell. The other role of the helper T cell is it interacts with a B cell. And the way that happens, let me just erase that line quickly. The way that happens is you'll have a B cell up here with its antibody. An antigen is going to bind. When that happens, the B cell is going then to present that to this helper T cell. So it's presenting, so this is like a little drawing of it too. It's presenting the antibody and the antigen bound together to the T cell. That activates the B cell and causes the B cell to replicate, and then causes the B cell to differentiate into two different types of cells. One of them is a plasma cell, and what the plasma cell does is it makes a ton of different antibodies that are specific to this particular antigen, and it starts dumping these antibodies out into the bloodstream by really the billions. And like you've had this experience where you start getting better all of a sudden, where you're really, really sick and you start feeling better. That's generally because you're getting all of these antibodies dumped into your plasma. And then the second thing it does is it forms memory B cells. So these are B cells now that are tuned to these antibodies. You make a bunch of them, you can store them in your spleen, and they're floating around, and now they can be recalled quickly. I'm going to show you that in this. This is just showing you the different things that the plasma will do. It's going to stay on the outside of the bacteria, prevent it from attacking. It's going to mark the bacteria or whatever it is for destruction by the macrophage, or they can lyse the bacteria. But I wanted to show you the memory component of this. So up here now, these are memory T cells, and then there are memory B cells here. So these cells are just floating around. They've already been activated by the T cell. They're waiting around to fight a virus. Whenever, or bacteria, whatever, whenever that second exposure to the antigen happens, these can immediately become cytotoxic T cells. They also then become memory cells again. So you're boosting up your response. So the speed of your response is much more quick upon being exposed a second time to a particular antigen, and the volume of, say, the antibodies that you're making are dramatically more. All right. So I hope that helps. I know that was quick, but my hope is that repetition of this material is